So thanks very much for the invitation. It's been really good to be able to think about Mendelian randomization briefly for a while for putting this together as opposed to COVID. So I'm going to just talk about what I see as the fundamental assumptions of Mendelian randomization. So this will be pretty basic compared to, I saw that Eric Chechen gave you a talk on Mr. Genius and related methods that he's been developing. So it'll be nothing like that. This is going back to some to the fundamental assumptions as I see it of Mendelian randomization. And it's a brief outline of the talk, which is to help me, not really to help you, but I'll just to say that I will end with a short story before finishing. Mendelian randomization, as I see it, is strengthening causal inference within observational epidemiological data through the incorporation of the special properties of germline genetic variants. And as the name implies, one might go back to Mendel to think of the justification of this, although in the form that we know it, in terms of Mendel's two laws that we all learned in basic biology, those are actually owed to Robert Heath Locke in 1906, because in Mendel's paper, which is a wonderful paper and highly recommended, but there are not the two laws put forward in the way that history has seen. And it was Locke who extracted from the papers the two laws that we know it. The first of Mendel's laws, which of course he developed in these cross studies with peas, was that as the genes are passed on, are passed down generations, the two laws are firstly that there's complete segregation of the two allelomorphs, as Locke referred to it, in equal numbers of the germ cells of a heterozygote. That constitutes the first and most important section. And what that means is that there's a segregation at the stage of gamete formation, and that in terms of trying to interpret what the causal effect of that, we must assume that that segregation is unrelated to, in humans, to the intrauterine environment or anything that happens during pregnancy, and that at live birth, that segregation has not been influenced by any distorting processes. And then we need to assume that that continues through to how people get into our study, to when people get into our study. And those things that now, with the data available in large population studies of mothers and offspring, like MOBA in Norway, you can check that that actually occurs down to live birth, and we can check that that then occurs through into our studies through quantitative analysis now. And the second part, or what we know as Mendel's second law, is that the separate pairs of allelomorphs segregate quite independently of one another. So that means that there's no genetic confounding. Of course, we know that there is linkage disequilibrium, that variants that are very close to the variants you're interested in will be associated with them, but outside of that, there won't be genetic confounding. So it's the first of the two laws which says that there should not be environmental confounding, and it's the second part of the law that suggests that there will not be genetic confounding. Now, the person who first considered using these principles in causal analysis was Sewall Wright, who was both a geneticist, but also is known today, of course, his major work was in genetics and in evolutionary theory, as well as physiological genetics, but he's also known as the progenitor of path analysis. This is his most famous early path analysis diagram, when he was looking at the coat color of offspring in guinea pigs. And in his path diagrams are an attempt to demonstrate causal processes diagrammatically. And beautifully in his work, he has a role of chance in the segregation of genetic variants. Those of you who've read Judea Pearl's brilliant book, highly recommended book, The Book of Why, will know that Pearl discusses Sewall Wright as a progenitor of the form of diagrammatic representation, but also of the form of a way of thinking that tries and interprets relationships in terms of causal effects. Now, Wright was, in developing his path coefficients, was completely explicit that what gave these path coefficients any form of causal interpretation was because of their anchoring through the Mendelian principles. Responding to a critique of his paper, Correlation and Causation, his 1921 foundational paper, he said it was of course realized that the concrete phenomenal actuality of the results wasn't proved by the analysis of the path coefficients. It wasn't the analysis that gave these any causal interpretation. This rests on the validity of the premises, i.e. and the evidence from Mendelian heredity. So it's these causal anchors that allows any such interpretation. And he said this again and again and again and again in different places. The hypothesis that heredity is Mendelian may usually be used safely as information external to a system. So that's your causal anchoring, which allows you to interpret the correlations as causal. Anyway, so this is something Wright repeated, and he would sort of turn in his grave, I think, seeing some of the ways which the descendants of path analysis in directed acyclic graphs are utilized without really without anchors, which give you this basis for causal analysis. Louis Gutmann, way back in 1977, pointed this out in a paper in which the subheading of one of the sections was "Path analysis does not analyze non-genetic paths," was that genetics has but one modest framework for paths. In contrast, according to current journals, sociologists keep discovering new fundamental path frameworks every month, and sociological graduate students are required routinely to hand in as individual class exercises new discoveries equaling Gregor Mendel's. This was a critique of path analyses were being critiqued in sociological and related sciences such as epidemiological science for not having this basis. And going back to Egon, Pearson and his famous correspondence with Jerzy Neyman during the development of the Neyman-Pearson lemma. In one of the letters, Pearson said that by 1929, I'd come down firmly to agree with Fisher that prior distributions should not be used except in cases where they were based on real knowledge, e.g. in some Mendelian problems. So the notion that these Mendelian laws actually gave you some basis for causal analysis was commonplace. But that actually being used in sort of epidemiological framework or in human health research, although there are progenitors where people were utilizing some aspects of the particular nature of germline genetic variation, and of course in the germline variation not as something actually measured or assayed as we do now, has been used, for example, in twin studies, which I'll get back to at the end, and other forms of study design that utilize this anchoring. But the actual term Mendelian randomization was first used in 1991 by Richard Gray and Keith Wheatley in a brilliant method they developed, which was only really applied years later because the data weren't available, for investigating whether bone marrow transplantation in some hematopoietic cancers, leukemias, lymphomas, etc., whether bone marrow transplantation was beneficial. It was very difficult to do randomized controlled trials in this area because, understandably, parents of children with cancers which at that time had very high mortality would want any treatment for which that might be beneficial to be utilized, and would not willingly go forward into randomization. And with bone marrow transplantation, what Graham Wheatley realized, or what was obvious, was that if a child had a sibling that was genetically compatible, was HLA matched, then they were much more likely to get a compatible transplant than a child who did not have such a sibling, because the siblings were very often were usually or very often the source of a successful compatible transplant. So they then went forward to say that therefore, if a child, whether or not if you went back and if a child who had one of these cancers and was a candidate for bone marrow transplantation, if they had a sibling who was compatible, then they constituted a group which together would be more likely to receive a successful bone marrow transplant. And of course, the compatibility of the siblings was entirely dependent upon the Mendelian laws. Hence, they use the term Mendelian randomization. And the model they put forward, as I say, they put this forward years before these studies were done, was to do a form of intention to treat analysis, which would look at all of the children with respect to whether their siblings were compatible, or they had a compatible sibling or not, and carried out that analysis. So that was the introduction of the term Mendelian randomization, but it wasn't used until the early part of this century outside of this framework. And then even within this framework, it was, I think there have been 15 papers or so referencing Graham Wheatley. But the advent of the human genome sequence and therefore the ability to develop studies where you were actually reliably identifying germline genetic variants which related to phenotypes allowed the same principles to begin to be applied in population-based studies. So Mendelian randomization, when Jerzy and I discussed this in 2003, we pointed out that it was clearly seen in parent-offspring designs. So when you had parents to offspring, or you had pairs of siblings, then there was out with meiotic drive or other now known to be, or even then known to be extremely rare, violations of the Mendelian laws. That was a form of real randomization. It's difficult not to see that as a process of randomization. The alleles being randomized from parents to offspring. And the analogy to RCTs that we mentioned was in relation to the parent to offspring design. And that in studies that were not familial, that were not based on the families, then the genetic variants were not strongly related to confounding factors in the population, something which has been empirically demonstrated again and again and again, but it was seen to be an approximate rather than absolute form of randomization. So Mendelian randomization currently is largely seen as implemented in an instrumental variable framework, although there are many examples, and this sort of work carries on, where the properties of genetic variants are used to strengthen causal inference in a way that is not translatable into an IV analysis. And I think that that form of strengthening inference through the particular properties of genetic variants is one which should be developed further, and should not be sort of abandoned because of the possibilities that exist in some of the areas for formal instrumental variable analysis. But this is the usual, this is the sort of picture or schematic that one sees for a Mendelian randomization approach. You have a genetic variant which relates to an exposure, say, circulating LDL cholesterol level. If you actually measure circulating cholesterol as a phenotype, then of course that could be confounded in its association with any outcome, be it coronary heart disease or cancer or whatever you're interested in as the outcome. And there will also be reverse causation, that the early stages of developing disease process will or can influence your exposure, and therefore generate a non-causal association between your exposure and your outcome. But because of the properties of germline genetic variants, they obviously do not suffer from reverse causation, and they are unrelated in general to factors that could be considered confounders, which I will come on to. So if your genetic variant relates to your outcome, then that strengthens evidence that the exposure influences the outcome. But now, if you want to make this interpretation about a modifiable exposure and therefore think about that as a target for modification, then you need to make what is the fundamental assumption in Mendelian randomization, which is that modifying the exposure through other means than through genetic differences, that the other factors which modify the exposure will have the same effect on the outcome as when the exposure is influenced by the genetic variant. So this is the fundamental assumption, is one of gene-environment equivalence, which has 100 years of being discussed in developmental genetics. And the examples of gene-environment equivalence come from Mendelian disorders, where there is a genetic variation that you know of that produces something which looks like something that is phenotypically produced. And that you can either call this phenocopy, if you consider the environmentally caused thing as copying the genotype, you call it phenocopy, or you can call it genocopy if you think the gene is copying the environment. It's just the way you consider this. The terms are identical. They're just facing the other direction. And an example of this is Hartnupp's disease. So Hartnupp's disorder was identified in the 1950s, and it resulted in a condition which looked very similar in terms of all of its symptomatology and the pathology as far as it could be examined. It looked very similar to pellagra. Now, pellagra was shown by Goldberger in the 1920s in his heroic epidemiological studies with others that you will know of, was shown to be due to dietary deficiency, and that dietary enhancement could prevent this condition, but it wasn't known which particular component of the diet it was within Goldberger's lifetime. But if Goldberger had had available to him the evidence from Hartnupp's disorder, which is caused by a mutation which leads to poor absorption of amino acids, including tryptophan, which is converted to niacin, then niacin would have been as the environmental factor causing pellagra. So this is the notion that it doesn't matter whether the deficiency is influenced by genetics or by environment, it produces the same outcome. So that is gene environment equivalence, which, as I say, is just commonplace in developmental genetics. Zucker-Candler and Villert, in a well-known paper many years ago, said no doubt all environmental effects can be mimicked by one or several mutations. Possibly not true for being run over by a truck, but for most external causes of disease, we can think that this will be the case. Or in developmental genetics, Mary Jane West Eberhardt refers to gene environment equivalence, gene environment interchangeability, the notion that something influencing gene expression will have the same downstream effects, whether that something happens to be genetic difference or it happens to be something environmental. So this is the assumption that you have to be willing to make if you think that the study is telling you about what is likely to happen on modification. So going back to our example of variants and LDL receptor genotype that that relate to circulating LDL cholesterol level, we've got to think that the differences in cholesterol created by diet or cholesterol-lowering medication, etc., will produce the same effect on the outcome as if that is produced by genetic variation. And thinking of the limitations of Mendelian randomization, this is the one we need to think of ways in which this when this won't occur, when this won't be seen. Now, in the case of cholesterol cholesterol lowering, the fact that for many of the drugs that have been tested in RCTs, randomized controlled trials, and reduce cholesterol and reduce coronary heart disease, those drugs target particular genes, which happen to be genes, which, well, don't happen to be, which of course are genes that influence cholesterol levels. That is HMG co-reductase for the statins, PCSK9 for the PCSK9 inhibitors, and Neyman pick C1-like-1 protein, which relates to transfer of cholesterol across the gut, cholesterol absorption, and that which is mimicked by ezetimibe as the drug, and will also reflect to an extent dietary differences, etc., and others. So in that situation, you think it's rather reasonable that cholesterol influenced by the genes and by these forms of factors that influence the distribution of cholesterol in the population will have the same effect. You can also, reduce people's circulating cholesterol levels by exsanguinating them, but they don't. But in the population, there are not genetic variants that relate to being exsanguinated. So there the equivalence would not apply. And if we think, if we replaced cholesterol with body mass index here, if we think of the now very large number of genetic variants and the huge amount of functional work about the activity of these genetic variants, then those genetic variants influence body mass index in the population through the mechanisms the mechanisms which relate to how the distribution of BMI in the population is produced. Those mechanisms include increased dietary intake, genetic variants influencing appetite, some forms of genetic, some variants influencing expenditure and energy loss, etc. I mean, there's a sort of cute example, which is a very nice example to use to think about the complexities of interpretation, about cutting someone's leg off would reduce their body mass index. Now that's true, but that doesn't. But of course, there's no genes for having your leg cut off. The genes influences the process, those processes which are related to the distribution of body mass index in the population. So the gene environment equivalence approach and thinking about whether it's reasonable or not also helps in thinking about what are actually sensible interpretations of the associations you see between phenotypes and outcomes. Here's just some data on genetic variants in different these are single genes that produce the protein. These are variants in those genes that relate to expression of those genes, and that these are targeted by drugs. So CETP is targeted by CETP inhibitors. This is in orange. I already talked about MPC1 like 1, and then HMG CoA reductase for statins, PCSK9 for PCSK9 inhibitors. Here we see the drug trials and genetic studies scaled to the same difference in HDL cholesterol. And you see what was predicted 20 years ago when discussing Mendelian randomization, when there was very little data, but there was some on cholesterol, was that, as you would expect, the genetic variants relate to long-term differences in cholesterol, whereas the drug trials last about 5 years. And we know that atherosclerosis is a process which develops over life. US soldiers in the Korean War, when they were autopsied at 18 and 19, already had atheroma. And now, of course, there's studies which show that these genetic variants relate to cholesterol in cord blood at age 4, at age 7, etc., etc. So you can look at the trajectories with these genetic, how these genetic variants influence LDL cholesterol from birth on. And as you would absolutely expect, just from the obvious biology of atherosclerosis, the drug trials, when you reduce cholesterol for about 5 years, produce about 35% of the effect that you would expect that is generated by long-term differences in cholesterol, and they line up rather well. So there's an example of gene environment equivalence for these genetic variants in relation to LDL cholesterol. So the instrumental variable assumptions, which are required for an IV analysis, are that the genetic variant is not independent of the exposure, which is the one which is testable. So the genetic variant exposure associations are tested, but then two non-testable assumptions. The first is of no confounding or exchangeability. Now, for genetic variants, the confounders, there's only a limited number of confounders. Remember, obviously, you can't change your germline genetic variant, so you've got to think of factors which influence the distribution of the variants and will independently influence the distribution of your outcome. And population ancestry differences, so population stratification, is the primary confounder, which in genome-wide association studies can be addressed with principal components and other forms of analysis, sensible restriction. The other forms of actual confounding will be factors that relate to selection into your study, can lead to distributional differences, and also the other form of environmental factor which is created is for some phenotypes, the parental genotype will influence the environment that the children grow up in, and that will lead to an association between the children's genotype and factors influenced by that environment. Now, these have now been studied. This has now been studied very widely, in very extensive samples, by looking between siblings, because of course siblings share the same familial environment. And what is seen is that the phenotypes which are influenced by this are ones that would be expected to be influenced by this are educational attainment, where the parental genes will influence for example, will influence the educational level of the environment, books in the house and factors like that will be influenced by the parental genes, and those genes are transmitted to the kids and will then also be associated with the environment created by parents. And there's some small effects on some behavioral factors, but for proteins and most phenotypes that have been examined within sibling, within sibship analyses are the same as the population analyses. But this is very important for examining, trying to utilize these methods, looking at social and behavioral factors, and in those need to be examined in depth. So that's confounding. Think of conventional confounding, but by far the most examined, at least, potential violation is that the genetic variants have an influence on the outcome, which is independent of the influence of those variants on your exposure of interest. So now this can actually be the single genetic variant, or it can be a genetic variant that is in linkage disequilibrium, i.e. that needs to be sort of closely physically related, but in the population, the genetic variants will be transmitted together. They probabilistically relate to each other because of proximity rather than because of population stratification. So this process in genetics is called horizontal pleiotropy, which is that the genetic variant has another pathway to the outcome. That's usually presented as, or Z is independent of Y, conditional on X and U. I mean, what it means is that there is an effect of the genetic variant on the outcome, which is not mediated by the intermediate phenotype that you're interested in. But that shouldn't be taken to mean that if you condition on X, that should remove the association. This is unfortunately, although it's done less and less, it was unfortunately thought of that was a sort of test of your assumption, was you adjust your LDL receptor variant effect on coronary heart disease for the cholesterol. But of course, doing that will introduce collider bias through leading to an association between the genetic variant and the confounders. So you introduce confounding by conditioning on the intermediate phenotype. So that shouldn't be done. So horizontal pleiotropy is obviously through phenotype. I mean, horizontal pleiotropy, genes don't have effects on outcomes that isn't meet, they must influence gene expression and then something. So there must be a phenotypic path between the genotype and the outcome. So this is horizontal pleiotropy. Probably commoner across the genotype phenotype map is vertical pleiotropy, which is when a genotype influences one phenotype, which in turn influences another phenotype, which influences the outcome. Now, vertical pleiotropy is not a problem for Mendelian randomization as long as you understand what is the primary phenotype. So, I mean, when you're looking at any genotype that's related to body mass index, for example, it must act through some other phenotypic pathway. But if you consider, for example, C-reactive protein, which is a very popular is a popular thing investigated by Mendelian randomization in the early days because it was a drug target and drug companies were developing ways of lowering C-reactive protein until the Mendelian randomization studies came around and suggested that C-reactive protein was not a causal factor. If you have all your genotypes that influence body mass index will also influence C-reactive protein, because higher body mass index leads to higher C-reactive protein. If you misinterpret those genotypes as being genotypes for C-reactive protein, it will of course appear that C-reactive protein causes coronary heart disease because body mass index influences coronary heart disease. So if you misattribute the primary phenotype, then that becomes a problem. Particularly problematic would be correlated pleiotropy, which is when the genotype influences some phenotype that you have not measured, may not know of, and that phenotype simultaneously influences your exposure and the outcome. In that situation, then the effects of pleiotropy will correlate perfectly with the effects of your genotype, or the association of your genotype with the exposure, which is something which violates the assumptions required from some of the sensitivity analyses, and that's just another form of correlative pleiotropy. So threats to the validity of Mendelian randomization are many. I mentioned some already. An important one in thinking from a developmental genetics perspective is canalization, which is that a genotype acting early in development, that the effects of that might lead to some form of developmental compensation to account for the phenotypic perturbation. Now, this is something which is seen in in major genetics disruptions. So that is certainly the case. I mean, you think with myoglobin, a sort of heart protein, if you generate a knockout mouse that doesn't have any myoglobin, then you actually get an alive mouse. I mean, it's very unlikely that this mouse isn't going to win the mouse Olympics, but the fact that it's born alive is remarkable, because if you imagined a thought experiment of sucking the myoglobin out of the heart of a mouse, you would have a dead mouse. So in extreme situations, this certainly occurs, although with the minor perturbations of the genetic variants that influence phenotypes like body mass index or LDL cholesterol, etc. It's not demonstrable, and the fact that you get agreements between trials and Mendelian randomization in so many situations suggests in these situations it's not coming into play, but it must always be thought about. And as was discussed right at the beginning at considerable length, the fact that the genetic randomization occurs at gamete formation and then conception, the fact that means that you have to think back to processes that act from that stage, and that you've got to, and it is the genetic effect from that stage which is the phenotype you are examining. You need to think about ways in which you can interrogate that. But pleiotropy has received by far the most attention, and I'll just spend sort of 3 or 4 minutes on sensitivity analyses that have been applied in this situation. The sensitivity analyses that have been used have generally taken the fact that there are large numbers of genetic variants that relate to exposures, and that it is unlikely that many different genes will have the same pleiotropic effects that perfectly balance out their effects on your outcome. The effects you're interested in on your outcome is vanishingly small. And so looking at the heterogeneity in effects of predicted effects is the primary way of interrogating these data. There are methods. I shall just give a list because that people can look up, but there have been many methods which have been developed. Egon was one of the first, which was rather than just regressing here, you see you regress the effect of the genotypes and LDL cholesterol against the risk reduction from coronary heart disease. Rather than forcing this through the origin, you let that float, and through letting that float, your slope still remains a valid causal estimate. This is like those of you who worked on LD score regression. This is the same principle as the floating intercept in LD score regression, but it depends on an assumption, which is that the effect of the genetic variant on cholesterol doesn't correlate with the pleiotropic effect, which with correlated pleiotropy will be violated. Now, there's a very large number of sensitivity analyses that have been produced, Some of these are central tendency, where they must automatically be true if the assumptions are true, such as the median estimator. So that the assumption with the median estimator is that if at least 50% of your instruments are valid, then the median instrument is valid. And that is the simple median estimator. And then you can use a weighted median estimator, which the assumption becomes more complex. And then there's a modal estimator, which is that simply the commonest estimate is likely to be the valid estimate, because it's unlikely that invalid instruments will all produce the identical estimate. But again, if it's correlated polarity, that can be the case. There's a lot that there's several central tendency estimators. There's estimators which exclude outliers, such as Mr. Presso, and there's estimators which have been developed to take into account correlated polarity, like Mr. Cause in Nature Genetics recently. There's a whole series of sensitivity analyses, including ones which use zero relevance points. These are particularly useful for behavioural factors like alcohol consumption, that the variance related to alcohol consumption in groups who don't drink shouldn't produce the phenotypic effect. So, for example, women in East Asian populations who would have very low levels of alcohol consumption in many large studies don't show the associations of alcohol consumption related variance with outcomes that would be that you see amongst men. You need additional assumptions in generating the actual IV effect estimates, which have been discussed at length. For example, homogeneity of exposure effect and outcome assumption, which assumes that your exposure is having the same effect on outcome across the whole population. That's quite a heroic assumption, but it allows you to it then allows you to call your estimate an average, a population average exposure effect, or the notion that the instrument affects the exposure always in the same direction, or has no effect or affects it in the same direction, which allows complier average of exposure effect to be estimated. This can actually be interrogated in a way going back to a suggestion of R.A. Fisher in 1938, which is that if he obviously didn't refer to this wasn't talking about it in these terms, he was talking about it in terms of interpreting trials in agricultural trials, and pointed out that if there was a heterogeneity of response, then for continuous outcomes, the variance would be inflated in the treatment group, for whom if some of the plants are growing taller and some are getting shorter because of the intervention, you would obviously increase the variance. And you can look at this with genetic variance. One thing Fisher said, he only introduced this in a letter to H.E. Daniels, but he said that he thought that the point about interaction, these heterogeneities were particularly brought out by people who didn't actually work in the practical experimental situation. And with genetic variants, you have huge amounts of data from studies like UK Biobank, which allow you to look at whether by variant, across the three genotype scores, the variance changes. And the bottom line is that there's rather little evidence of variance inflation. And in fact, rather beautifully, when you pick out the top example of variance inflation in UK Biobank, looking at very large numbers of phenotypes and very large numbers of genotypes, is one which is a beautiful Mendelian randomization example. It's that a genotype related to cigarette smoking, the genotype that's related to heavier smoking has a much higher variance for lung function measures than the genotype related with less heavy smoking, because of course the people who aren't smoking within those genotype groups but there will be will not have a variance difference, but the ones who smoke, the ones who are smoking heavier and heavier and heavier, who are smoking heavier, have greater effect on the lung function. So I realize about rush. I'll rush very quickly. Interpreting IV estimates, you need to be careful about thinking about the length of time of exposure, as I've talked about. Might only relate to exposure during critical period. We have a good example of that with vitamin D and multiple sclerosis, where the Mendelian randomization suggests vitamin D is protective, but when you triangulate it with other epidemiological evidence, then the likely story is that it will only be vitamin D period of exposure to Epstein-Barr virus. It can relate to an unmeasured upstream phenotype. So if you're taking your genetic variant to be a variant for, as I said, with the vertical pliotraphy example, a downstream phenotype, you make the wrong interpretation. It might not relate, it might only relate to a subpopulation of interest, might only be interpretable in terms of the liability to the exposure, particularly for categorical exposures, not the exposure itself, which I think is an important point, because many Mendelian randomization studies have been done which have said that they are demonstrating that a disease, for example, type 2 diabetes, causes coronary heart disease, but in a situation where the people in your sample, many will not have actually have had coronary heart disease. So in these situations, it's likely that the liability to the condition rather than the condition itself, which influences the outcome. And these are the situations where, when you have no relevance point, you can actually separate the liability from the actual categorical condition. And most importantly, as genetics is being used to attempt to understand treatment effects, the fact that nearly all our genetic data actually relate to disease occurrence, like, have you got this cancer or not, and you do a GWAS of having cancer, not to the outcome after developing the disease, is a massive hindrance, because for most conditions, or very many conditions, factors that influence the disease onset, such as smoking and lung cancer, are not good targets for treatment. Stopping smoking after you develop lung cancer is not a useful approach, is not a useful outcome, is not a useful way of dealing with lung cancer. So what we really need are genetic studies of disease progression, when Mendelian randomization could actually help generate evidence about treatments. So why generate IV estimates? Well, the IV estimates, given all these problems of interpretation, you need them for the sensitivity analyses. Comparing them with what you see in randomized trials can help tell you about immediacy, relative immediacy of treatment effects. In some cases, you can date them, such as smoking during pregnancy. I shall skip this. This is just to say that there's real problems with two-sample Mendelian randomization, which is extremely easy to do. I'll say that now, finally, we now have data which allows within family Mendelian randomization to be done, Mendelian randomization within SIBSHIPs, which is protected from dynastic effects, from population stratification, etc. And I think these data sources will become the very strong sensitivity analysis for Mendelian randomization. And I'll finish my story, which I'll do in 3 minutes, I promise. 2.5 minutes. My story is just to say, I started an epidemiology in the early 1980s in the Medical Research Council Epidemiology unit in South Wales, where they were setting up a study to study the effects of HDL cholesterol and coronary heart disease, because this was really about the hottest topic in, this is Archie Cochran doing the smoking and doing the Times crossword. And I went out and took blood samples in this study 30 odd years ago. We did detailed lipid challenge tests were being done to study this issue. A paper by Hulley had come out saying that the association between triglyceride and coronary heart disease was because of confounding, I think, which is by HDL cholesterol. Triglycerides were told, don't measure it, it's dropped. In 1991, I did some work with Andy Phillips, where we looked at the triglyceride HDL cholesterol story using data from British Regional Heart Study, and we demonstrated in the Regional Heart Study the story that Hulley found, which was that HDL cholesterol dominated the analysis. After adjustment, the triglyceride effect attenuated. But we just did some very obvious forms of sensitivity analysis, showing that when I took the blood sample, when he was involved in a blood collection, triglyceride samples, once you've got your spun down sample, if people had had 5 pints of Guinness the night before, you could see it, because triglycerides are massively variable and had considerably more variation than HDL cholesterol. And we showed that if you assumed that the interclass correlation for triglycerides was 0.6 and for HDL was 0.9, the model would reverse. HDL cholesterol effects would attenuate dramatically. But if you change that to a very small degree, model would flip back again. So this just suggested that this was not reliable. The epidemiological data were not reliable. But studies were carried on being done of HDL cholesterol, and large pooling projects appeared. And the triglyceride effect here is shown just to disappear on HDL cholesterol adjustment. HDL cholesterol effect is robust. And 2009, still it was concluded that HDL cholesterol may generate substantial additional benefit if targeted. So this is actually saying this would be a target for treatment trials. But by then, trials were being done. The first Mendelian randomization study came out in 2006, suggesting that this was HDL cholesterol did not appear causal, just looking at circulating HDL cholesterol levels. And then later, large-scale Mendelian randomization studies utilizing multivariable Mendelian randomization methods. I'll get to the end here. That suggested that HDL cholesterol is not causal. Now, this is something which the trials have shown, and now a very large number of trials with many different methods of modifying HDL cholesterol through different pathways, I think would rule out the notion that circulating HDL cholesterol, just thinking of it as that phenotype, that modifying your circulating HDL cholesterol in itself is not going to reduce coronary heart disease, even though literally billions of dollars are spent on randomized controlled trials of HDL cholesterol. There's a patent from Harvard which is on HDL cholesterol. Hopefully it's not on modifying circulating HDL cholesterol. Now, this isn't going back to when I started. I started a bit after this photo was taken, but going back to when I started, I think now, I think that conventional epidemiology, which at that time was studying many of these sorts of factors like fibrinogen and coronary heart disease and etc., and other clotting factors, that I don't think conventional epidemiology can get you close to causality with those things. I don't think there's any conventional epidemiological approach which was casting doubt on HDL cholesterol. There weren't, and I actually think the domain of epidemiology for studying these processes for things which one might want to modify. You can modify these things not just through pharmacotherapeutics. There are other ways of modifying such factors. Is not going to make a contribution. Deciding whether the key factor finally influencing atherosclerosis is ApoB or is other factors is not going to be advanced by conventional epidemiological studies. And I think the integration of biology and particularly the very particular characteristics of germline variation is key. But this must be put in a framework of triangulation with other evidence. So the fact that, I mean, the Mendelian randomization studies on many actual processes in lipid biology and in clotting and inflammatory systems, have come up with evidence which would certainly is influencing, stopping basically pushing forward trials and pushing forward trials in other areas. But the evidence must be triangulated, and there's a forthcoming collection, Cold Spring Harbor Perspectives, on integrating human genetics with causal inference methods to understand human disease. But Mendelian randomization will be one prong of those forms of the evidence that can be triangulated. And that's it. Thank you.